G2P-SOL is an acronym that means transforming genotypes of solanaceous plants into phenotypes, G2P. Solanaceous plants are very important for mankind. They count very important food plants like potato, which is the staple food for 8 million people around the world.
The four crops, the tomato, potato, eggplant, pepper, are all solanaceous species, so they are from the same family. And what is unique about them is that they are sharing many of the genes, much of the genetic code that allows them to develop and be the crops that they are.
In Ireland in the 19th century there was a big famine because a potato pathogen arrived to Ireland, and the varieties that were moved to Ireland from South America did not carry any resistance to this pathogen because they had never seen this pathogen in their previous life. So there was this complete wiping out of the potato crop, and a lot of people died of starvation. And G2P-SOL aims at restoring the complexity and the richness of the original germplasm of these four crops.
So there are a lot of genetic resources, tens or hundreds of thousands of accessions that are stored, but the information that regards these varieties is not accessible.
Genotyping by sequencing is a new genotyping method which has been developed in the last 5 years, which involves actually using directly the sequence of an individual's DNA as a marker. This was not possible until very recently. In G2P-SOL, we have decided to focus mainly on the use of genotyping by sequencing.
In fact, these two projects have something in common, which is TomGEM works on just one of the four crops, tomato. We work on all four. The second point in common is that both projects want to retrieve germplasm varieties that are resistant to stress. TomGEM is focused on heat stress. As you know, heat is an ongoing problem with climate change. We are more broad in our vision. We work on more crops and we work on more types of stresses, but there are certainly some points of possible collaboration. That's why we decided to make a common meeting.
For tomato, one of the big problems is for fruit yield at high temperatures. So the plants grow very well, but they don't make enough fruit at high temperatures. So we need to select varieties that have a greater tolerance in terms of their fruit yield and their fruit quality at high temperature.
Why do we look at genes? The reason we look at genes is because scientists believe, and there's a foundation for this, that when some trait, for instance, that the ability to grow under high temperature is inherited, it's because it's a genetic basis. So there's a gene behind this, a gene or several genes. So the way we are going to address this is we are going to select varieties that have this ability to grow under high temperature, and then to do crosses in order to generate population that will allow us to identify the genes behind this high tolerant trait.
TomGEM is unique in the way that it gathers complementary expertise from academics and non-academic partners that bring their know-how, they bring their resources, they bring their methodology, and converge, all those they converge to address a single equation, which is yield issue in condition of high temperature. The non-academic partners are essential in the TomGEM project because they include not only breeders, but also tomato producers and growers, and also biotech companies that are involved in evaluating the quality of the tomato crops. The outcome of this project will be hopefully beneficial to the consumer in different ways. First, it will hopefully ensure, and maintain yield in harsh conditions, but also it will maintain high quality, sensory and nutritional quality of the fruit, and this is of course directly for the benefits of the consumers.